Hello, students. I'm Byung-il Bae, an assistant professor of neuroscience. I'm sorry that I cannot be with you today. Regardless, I hope that this recorded video will help you to understand what we do in the lab. This is the coronal section of the human brain, and the cerebral cortex is the largest part of the brain, occupying more than 80% of brain volume with tens of billions of neurons. This large unfolded cortex is what makes us who we are as a species and as individuals. This is why we humans have amazing intellectual powers and also why we suffer so much from neurological and psychiatric disorders such as autism spectrum disorder and schizophrenia. To investigate cortical development, we leverage the neurodevelopmental gene ASPM, which stands for Assembly Factor for Spindle Microtubules. In humans, loss of function mutations in ASPM are the most common cause of abnormally small cortex or microcephaly. Furthermore, rare variants of ASPM have been implicated in autism spectrum disorder. It encodes a centrosomal protein that is conserved in all animals, including worms and flies. ASPM contributes to formation of the microtubule organizing center during metaphase and the basal body for the cilia during interphase. Loss of function mutations in ASPM significantly decrease proliferation of cortical progenitors and increase their premature differentiation, thereby limiting the size of progenitor pool. Interestingly, ASPM also shows a strong signature of positive selection in the lineage of animals with large unfolded cortices and is therefore more conserved between humans, non-human primates, and carnivores, and less conserved between humans and mice. Thus, ASPM is a molecular handle for us to address multiple aspects of cortical development. Currently, we have two projects in the lab. The first project is neurodevelopmental basis of social cognitive behavior. Nearly 20% of young patients with autism spectrum disorder have abnormally enlarged cortex or macrocephaly from a few months after birth until late adolescence, especially in the prefrontal cortex. But how macrocephaly underlies autism is unknown. Interestingly, ASPM variants or missense mutations have been found in autism. We wondered whether some of the autism-associated variants might actually be gain of function, and indeed, we found one mutation, alanine 1877 threonine, significantly increased the protein levels of ASPM, suggesting gain of function. Next, we generated knock-in mice carrying the gain of function mutation, which demonstrated macrocephaly with thicker cortical layers compared to wild-type animals. Most importantly, the knock-in mice demonstrate social deficits in three-chamber assay in a male-specific manner. This is significant because autism is 4 times more common in males than females. Therefore, we have a unique mouse model to investigate the causal relationship between macrocephaly and autism. To investigate the mice further, we are currently analyzing them using transcriptomics, proteomics, neuroimaging, and electrophysiological/circuit analysis through collaborations. The second project is unique vulnerability of gyrencephalic brain to neurodevelopmental disorders. Humans suffer from severe anatomical and functional phenotypes of brain disorders, but mouse models often show mild phenotypes. For example, loss of ASPM causes dramatic microcephaly in humans and ferrets, but mild phenotypes in mice. The species-specific severity is not only anatomical, but also cellular. Only in knockout ferrets, but not in mice, premature differentiation and delamination of cortical progenitor cells is observed, as shown by immunohistochemistry of the germinal zones. What is the molecular mechanism of gyrencephalic brain-specific vulnerability? We address this question by examining ASPM interacting proteins in the developing mouse and ferret cortex. Here we use a powerful proteomic technique called proximity labeling or biotinylation by antibody recognition. The rationale is that human and ferret-specific ASPM interacting proteins underlie the unique vulnerability. As species specificity is such a pervasive problem in brain disorders, we believe that proteomic comparison of mouse and ferret and human brains will be a powerful method to reveal molecular mechanisms. Currently, we're a lab of 4 people, postdoc Sonu, technicians Holden and Mushura, and myself. The only missing component is graduate students, so I'm eager to take on students. Our autism research is funded by NICHD and Eagles Autism Foundation, and our geroencephalic development research is funded by Brain Research Foundation and Yukon Research Excellence Program. We are fearless, friendly, and caring people. I'm sure that you will enjoy working with us. So please come and visit us. Thank you.